Hello dear students, today I will continue the chapter Molecular Basis of Inheritance. In the previous classes we have studied about the chemical composition of nucleic acids, the nucleosides, nucleotides followed by the polynucleotide chain, and then we have discussed about the structure of DNA, that is a double helix model of DNA, and followed by the packaging of double helix in the prokaryotic cells as well as in the eukaryotic cells. So now we are going to study about the various experiments related to molecular biology, the search for genetic material. As we all know, DNA is considered as genetic material, but how it was discovered as genetic material, how it was proved as genetic material. The beginning of such experiments was done by Griffith, so that is called as Griffith's experiment, and followed by Avery's experiment, and finally we will discuss about Hershey and Chase experiment. So these are the three important experiments which will be discussed today in this video. Let me start the first one, that is Griffith's experiment. Say Frederick Griffith, he was actually studying about the pathogenicity of the bacterium called as Streptococcus pneumoniae. You know, Streptococcus pneumoniae is responsible for the pneumonia disease. It causes pneumonia. In 1928, Frederick Griffith, in a series of experiments, identified the transformation in bacteria. So transformation is a special kind of phenomenon found in the bacterial cells. It is actually a kind of parasexual mode of reproduction. And while studying the pathogenicity of the bacterium, he identified two strains of bacteria. One is called as S strain, another is R strain. S strains are the smooth strains. These are encapsulated bacteria. Encapsulated means externally, the cell wall is externally covered by an additional protective layer called as capsule. And S strain cells are pathogenic, but R strains or rough strains are non-pathogenic because they are not externally covered by the protective layer, so they can be easily destroyed by the immune system of the host cells. Let me explain the steps involved in Griffith experiment. So he used the mice for his experiment. So the experiment was conducted in a natural condition. So that's why it is also considered as in vivo experiment. So he used mice for his experiment. The mice infected with S strain or virulent cells was died. So he injected the S strain bacterial cells into the mice, and such mice was died because of pneumonia disease. But when the mice infected with the R strain bacterial cells, they did not develop any pneumonia. That means the R strains are non-pathogenic. R strains does not cause the disease. The name of the bacterial cell is Streptococcus pneumoniae. But all the strains of Streptococcus pneumoniae are not able to cause a disease. So only the S strains are pathogenic and R strains are non-pathogenic. Actually, this was the experiment of Griffith. See, in the first step, he collected the S strain bacterial cells, and he took such S strain bacterial cells in a syringe. And then he injected such S strain bacterial cells into the mice, living mice. When the S strain cells were injected into the mice, normally the pathogenic bacteria causes disease that resulted in the development of pneumonia in that mice, and that caused the death of the mice. In the next step, he took the R-strain bacterial cells. He took such R-strain bacterial cells in a syringe. Then he injected such R-strain bacterial cells into the living mice, another mice. But when the R-strain bacterial cells were injected into the mice survived. So there was no death of such mice which was infected by R-strain bacterial cells. So by these two steps, we can come to conclusion that only the S strains are pathogenic that resulted in the death of the mice. But R strain bacterial cells are non-pathogenic that will not cause any harm to the mice. But he continued his experiment. In the third step, Griffith was able to kill the bacteria by heating them. That means he killed the bacterial cells, particularly the S strain bacterial cells, which was pathogenic, which was able to cause a disease to the mice. So he killed the S strain pathogenic bacterial cells by heating them, and then he injected, see, this is the S strain cells, such S strain bacterial cells were killed by heating. You can see the boiling, and then he collected such S strain bacterial cells, that is heat killed S strain bacterial cells in the syringe. Then he injected such a heat killed S strain bacterial cells into the mice. As we know, as we observed in the previous steps, S strain bacterial cells are pathogenic. But here in this step, the difference is such S strain bacterial cells were killed by heating. Then he injected such heat killed S strain cells into the mice, and such mice survived. So there was no development of pneumonia disease in that mice. So what it says? It says that when the pathogens are killed, when the pathogens are inactive, such dead pathogens are not able to cause the disease. He continued his experiment. So in the next step, he injected the mixture of heat killed S strain cells as well as living R strain bacterial cells into the mice, and the mice died. Moreover, he recovered living S strain bacterial cells from the dead mice. Just observe here. These are the R strain bacterial cells. And you know, R strain bacterial cells are non-pathogenic. They will not cause any disease. He mixed these non-pathogenic R strain bacterial cells with heat-killed S strain cells. Again, heat-killed S strain cells are also non-pathogenic. So he got a mixture of two non-pathogenic bacterial cells. Even though R strains are living, they are non-pathogenic. But here pathogenic S strain cells were killed, so they becomes non-pathogenic. He took the mixture of non-pathogenic R strain cells and non-pathogenic heat-killed S strain bacterial cells in the syringe, and then he injected such mixture into the mice. After injecting into the mice, he noticed the death of the mice. In the next step, he recovered the S strain bacterial cells from the blood of dead mice. So it was totally different. He never expected that. So what is the reason for this? He came to conclusion that The R strain bacteria had somehow been transformed by the heat-killed S strain bacteria. So some factors found in these heat-killed S strain bacterial cells are responsible for the conversion of R strain cells into S strain cells. So some transforming principle transferred from heat-killed S strains had enabled the R strains to synthesize a smooth polysaccharide coat and become virulent. So this was the conclusion given by Griffith. But he never told about the DNA. He said that some factors found in the heat-killed S strain cells are responsible for the bacterial transformation. So this must be due to the transfer of genetic material. But what is that genetic material? It was not informed by Griffith. So that's why the Griffith's experiment was continued by some other scientist. However, the biochemical nature of genetic material was not defined from his experiments, and that was defined by some others' experiment. Now let me move to the next one. The biochemical characterization of transforming principle. So this is a continuation of Griffith's experiment. Nothing but the second experiment, that is Avery's experiment. So prior to the work of Oswald Avery, Colin MacLeod, and Maclean McCarthy, the genetic material was thought to be a protein. These three scientists, they worked to determine the biochemical nature of transforming principle in Griffith's experiment. So what they did in their experiment? They purified the biochemicals, that is proteins, DNA, RNA, etc. from the cells, from the heat-killed S strain cells, to see which ones could transform living R cells into S strain cells. They never knew about the specific chemical component responsible for the bacterial transformation. But they had a doubt on proteins, DNA, RNA, and some other chemical components. So that's why they isolated such chemical components from the heat-killed S strain cells. And they discovered that DNA alone from its bacteria caused R bacteria to become transformed. So by this, their experiment, they discovered that DNA alone from S bacteria caused R bacteria to become transformed. Then how they identified it? In their experiment, they used some certain enzymes to remove the specific chemical component, biochemical component from the mixture. In the first step, they removed the proteins by using proteases, but they got S strain cells even in the absence of proteins. In the next step, they used RNA digesting enzymes called as RNases to remove the RNA, but when they mixed the living R strain cells and the chemical components without RNA, they got S strain cells. In another step, they took the mixture of living R cells and the heat-killed S strain cell chemical composition without the DNA. They got only R strain cells. They never got S strain cells. And they used a specific enzyme called as DNase to remove the DNA from the mixture. Say, when they used proteases, they removed the proteins, they got S strain cells. When they removed RNA molecules by using RNase, again they got S strain cells. But when they removed RNA from the mixture, they did not get S strain cells. By this, they concluded that DNA is responsible for the bacterial transformation. They concluded that DNA is a hereditary material, but not all biologists were convinced by their experiment. So, two more scientists conducted one more experiment. Alfred Hershey and Martha Chase conducted an experiment to know about the specific genetic material. So in their experiment, they used a special kind of medium as well as a special organism for their experiment. They used bacteriophages for their experiment. The unequivocal proof that DNA is a genetic material came from the experiments of Alfred Hershey and Martha Chase in 1952. They worked with viruses that infect bacteria called bacteriophages. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that enter the bacteria. If the protein enters into the bacteria, that should carry the genetic material. If the DNA enters into the bacteria, that should be responsible for the transfer of genetic information. So they grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. That means they culture the bacteriophages into different types of medium. One medium with the S35 radioactive sulfur and another medium with the radioactive phosphorus P32. Viruses grown in the presence of radioactive phosphorus contain radioactive DNA because phosphorus is found in DNA, but not radioactive protein because protein does not contain phosphorus, because DNA contains phosphorus but the protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA because DNA does not contain sulfur but the protein consists. So they got two types of bacteriophages. One group of bacteriophages with radio-labeled protein coat, unlabeled DNA. Another group of bacteriophages with radio-labeled DNA, unlabeled protein coat. So radioactive phages were allowed to attack, not attach, or attach, that is also right, because after attachment to the bacterial cells, they will enter into the cytoplasm. So radioactive phages were allowed to attack to Escherichia coli bacteria. Then as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. You know about the viral infection. During viral infection, the entire viral particle will not enter into the host cell. Only the genetic material will be introduced into the host cell. And the empty protein coat will remain outside the host cell. That empty protein coat without genetic material is called as ghost. And after the completion of infection process, they separated the viral particles from the bacterial cell surface by the process called as agitation. Blending, that is also called as blending. The agitation means shaking by using the instrument called as blender, and the process is called as blending. And after that, the viral particles were separated from the bacteria by spinning them in a centrifuge. So when the bacteria which was infected with the viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. I repeat, bacteria which was infected with viruses, that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were non-radioactive. radioactive. When the radioactive property was found in the protein coat, the bacterial cells infected by such kind of bacteriophages did not get the radio label or radioactive property. So what it says, this indicates that proteins did not enter the bacteria from viruses. DNA, therefore, the genetic material that is passed from the virus to bacteria. It was a conclusion. Here you can see the diagrammatic representation. So this is a bacteriophage cultured with the S35 medium. These are the bacteriophages cultured in P32 medium. P32 medium. Phosphorus will be there in the DNA. So here the DNA is radio labeled, and here the protein coat is radio labeled because sulfur is found in the protein coat. And such bacteriophages are allowed to infect the separate bacterial cells. When the bacteriophages cultured in the S35 medium are allowed to infect the bacterial cells, the genetic material, that is DNA, entered into the host cell, but radio-labeled viral particles remained outside. And on agitation, they got non-labeled bacterial cells and radio-labeled bacteriophage protein coats, viral particles. But in another case, when they allowed the infection by the bacteriophages cultured in the P32 medium, they got radio-labeled bacterial cells. So here, the radio-labeled property of virus was transferred to bacteria. That is because of radio-labeled DNA. So by this experiment, they concluded that only the radio-labeled property can be inherited from virus to bacteria if it is found in the DNA, and it will not be inherited from virus to bacteria if that property is found in the protein coat. So by this they concluded that DNA is the genetic material. Here one animation is given. So this explains Alfred Hershey and Martha Chase experiment. Just watch it.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell.
Say this is a normal procedure. Here this is the bacteriophage with the protein coat and the genetic material, and normally here the protein coat is radio labeled because this bacteriophages was cultured in the S35 medium and the DNA are unlabeled. When these bacteriophages with the radio labeled protein coat were allowed to infect the bacterial cells, only the genetic material introduced into the bacterial cells, and such genetic material resulted in the production of many number of bacteriophages, and such bacteriophages were unlabeled. Bacterial cells did not get the radio label property, and this is a bacteriophage cultured in P32 medium. When the bacteriophage is cultured in the P32 medium, the DNA will get the radio label property P32. Such bacteriophages are allowed to infect the bacterial cells. Genetic materials are introduced into the host cells, and such radio labeled viral particles result in the production of viral particles and the viruses. So here the bacterial cells got radio labeled property, and the removed bacterial viral particles were unlabeled. So by this experiment, they concluded that DNA is a genetic material, but not the protein molecules. So now let me tell you about the important criteria that must be fulfilled by a molecule to act as a genetic material. One of the very important topic. The first point, to become a genetic material, that molecule should contain these important properties. First one, it should be able to generate its replica, and by the process called as replication. Only DNA is self-replicable. From one RNA molecule, another RNA molecule can be produced, but it has to undergo reverse transcription followed by transcription. It cannot produce another RNA molecule directly, but DNA is self-replicable. The another point, it should be stable chemically and structurally. When compared to RNA, DNA is more stable. The another important property, it should provide the scope for slow changes, that is called mutation, that are required for evolution. Of course, DNA is capable of undergoing mutation. Even RNA can also undergo mutation. And the last one, it should be able to express itself in the form of Mendelian characters. Of course, DNA is self-expressible, even RNA also. But still, DNA is considered as genetic material, even though the RNA also consists almost all these important properties. DNA is considered as genetic material. Why? So, answer to this question will be given in the next video. So, I will stop this chapter here. The remaining topics will be covered in the next video. Thank you.